First of all, I would like to welcome all of you to the IAS Distinguished Lecture. It is our great honor to have Professor John Lehn come here today to give us a talk on from matters to life chemistry. Okay. Yeah. Since I'm also trained as a synthetic organic chemist, right? So I really applaud for this title. Professor Lehn is an old friend of CTU and a senior fellow of IAS. He and some of other senior fellows witnessed the inauguration of IAS in November 2015. IAS is one of the few such centers in the region. It provides our faculty and students with an exciting environment to advance their work, and also serve as a catalyst for many of the aspirations articulated in our strategic plan for 2015 to 2020. This week, in particular, is a very special and also auspicious to CTU, since we have 3 Nobel laureates visiting us, namely Professor John Lehn, Professor Serge Harash, and also Professor David Winland. Both Professor Lehn and also Professor Harash are our IAS senior fellows, and we hope that Professor Winland will join us in the CTU family in the near future. There are 2 major events this week involving these 3 distinguished scholars, including today's lecture and also the IAS workshop on quantum science to be held on November 8th and 9th, making it a Nobel Science Week. Last year, Professor Lehn brought along quite a few outstanding scholars in the field of chemistry to CTU by organizing an IAS symposium on chemistry of the complex matter. The CTU community has benefited greatly from the workshop. We are truly grateful to Professor Lehn, and we are very happy that Professor Lehn comes again, and this year to continue to share with us his valuable thoughts and also insights. Before the lecture, may I invite Professor John Lu, Vice President of CTU University of Hong Kong, to give a brief introduction of Professor Lehn. Professor Lu, please.
Thank you, Professor Alex Chen. Good morning, Professor Jean-Marie Lehn, honorary guests, colleagues, friends, and fellow students. It's my great pleasure to introduce Professor Jean-Marie Lehn today. As Alex mentioned, Professor Lehn is an old friend of CTU University of Hong Kong and has strong connection with us. He joined CTU in 2008 as chair professor at large and has been visiting distinguished professor of Department of Chemistry since 2013 and a senior fellow of IAS since its inception in 2015. In 2010, CTU conferred an honorary doctorate to Professor Lehn in recognition of his invaluable contribution to our understanding of chemistry and the world we live in. Regarded as father of supramolecular chemistry, Professor Jean-Marie Lehn's innovative research was the promise for an entire new field in chemistry, and today play a central role in numerous disciplines. His research accomplishment has earned him more than 100 awards and honors, including the 1987 Nobel Prize in Chemistry and numerous fellowships of Science Academy worldwide. He has held teaching positions at a number of distinguished universities and has been a professor since 1979. Besides lecture around the world, Professor Lehn is now the director of the laboratory supramolecular chemistry at his alma mater, Université de Strasbourg. Without further ado, may I now invite our distinguished speaker, Professor Jean-Marie Lehn, to the stage to deliver his lecture entitled From Matter to Life, Chemistry, Chemistry.
That's a very full room. You know, that also means that IAS is really making an impact. Something like the IAS, an Institute of Advanced Study, at the beginning you wonder whether it will be useful or not. When I see this room, definitely it will be very useful. It's a new institution. It has developed very quickly. And last year, there were much fewer people than this year, which means that one can hope that in the future it will develop even more. And I'm impressed. Thank you all for coming. I'm also impressed because our famous physicist in the front row. So I'm very afraid, I must say. Serge says I look like afraid. But we are good friends. He will not be so bad to me. So it's nice to be back here at CTU to see also the evolution of CTU. I have been coming here first on the invitation of Philippe Sierlet, a mathematician, a very famous French mathematician. And so it's nice to continue that. He has had the good idea to invite many French scientists, including Claude Cohen-Tanouji, for instance, and some others. So it's a pleasure to be back and to tell you about some aspects of chemistry. I'm very, very thankful to this high-brained physicist to come to listen to a chemistry lecture. So, yeah, when you give a general public lecture, this is a general public lecture, and young people from the international school here, first I have to have a title which can attract people. So from matter to life, that looks fantastic, huh? Everybody's excited. Then you add chemistry. Huh. People wonder, what is this guy doing? Talking about chemistry in life with a question mark. And in addition, yes, as the provost said, chemistry. He's a chemist. So, yes, I would like to show you not how you go from matter to life, because this we don't know yet, but some steps along the way. And for that we have to start very early at something which physicists like, here. It all started, physicists tell us, what can we say? We have to think that they are right, with the Big Bang. At the Big Bang there was no chemistry. It was only physics, if you will, if you wish. That's the age of physics at the beginning. The universe is very hot, energy, very hot, nothing else. Then particles formed. Particles formed, you see, for instance, in 1 second, or the other one is better, 1 second here, then 3 minutes, and then particles formed and they began to get together to form more complex entities, atoms. And chemistry started around here, when atoms were formed, and they could then lead to more complex entities, like from bricks to make a house, from atoms to make an entity called a molecule. That's the age of chemistry. These molecules evolved progressively. We'll have a little bit a look into how this could evolve. And they formed larger and larger molecules, which had some features which make them aggregate, define compartments. And these compartments were the beginning of a primitive, what you might call a cell. And then at some stage, a property appeared on our planet Earth, which is life, about at that time. That's the age of biology. It happened that it didn't stop there. Living entities evolved to more and more complexity, and at some stage another property appeared. In fact, for me, much more important than life itself, which is that this entity formed on our planet Earth is able to think. And we are here because of that. Otherwise, we would not be here. Now, one comment first. This is the end of the screen. But that is not the end of evolution. We as human beings are just a point in space and in time. So we have to be modest. On the other hand, we can be very proud. This thing we build up in understanding. We are far from knowing everything, but being able to think about the origin of us and of the universe is something quite incredible. In fact, it's for me a question which is a very difficult one. How is it possible that the universe could generate an entity able to think on its own origin? I don't know, but it happened. Now, this is not the end of evolution, and nowadays there are a number of people who think that we should stop where we are. This is totally impossible. We will not stop. The universe will continue to evolve, and we will change. We will even change ourselves. We come back to that at the end. And in other regions of the universe, there is, you can never say it's 100% certain, 100% does not exist, but it's highly probable there are many forms of life in many regions of the universe. It's so big, but the probability is in favor of life and not against it. So that's from matter to life. Now, if you want to characterize a little bit very schematically, very simply, the evolution of matter, if you take something like the complex, how complex the object is, how much, so to say, information is contained in it, we may say that we start simply with divided matter. This divided matter becomes then condensed, it gets together, but that's not enough to be condensed. It can't organize. How? On the basis of given features. Then it has become living at some stage, then it has become thinking. Beyond that, there may be something else. Our way of thinking, is there something else? We don't know. We cannot exclude it, that's all we can say. And maybe there are something even more powerful ways of behavior, features of higher complexity, you may say, than our own thinking. This is a sort of evolution towards complex matter, and one might say that it looks like it's a sort of a pressure of information. Things get more and more complex and informed in given regions of the universe. Now, it's a bit talking about pressure of information. It's like a sort of using Darwinian type of wording. There's a pressure of something which pushes things in certain regions towards more and more complex entities. At that stage, we have to ask a very big question. In fact, despite the presence of famous physicists, the big question is what? How does matter become complex? Because this is how an elementary particle can come to a thinking organism, maybe even to higher, more complex forms of life. And you know, what is important, of course, we know that, let's say, since they're here, quantum physics is fantastic, changed the world. But on the other hand, when Einstein also, he proposed these laws, which now have been confirmed, but by the experiment on gravitational waves, the question is, how could Einstein exist? How could the universe produce an entity which is able to generate these features, this thinking? And for doing that, to answer this question, mankind has created something very important, which we call science. In science, physics studies the basic laws of the universe. We all depend on that. These are the laws which run everything. And we all depend on it. That's the basis. Then biology tries to understand the rules of life. How do we function? How do living systems function? We know that it is very complex. We have to understand it. It exists, therefore it's possible. But it doesn't tell you how. And then what is chemistry doing? Chemistry, I think, has this very important mission to try to build a bridge between the basic laws which run matter and the way in which these laws can be expressed in a given feature, in a given entity. It can be a thinking organism, like we are, or other types. What's the way? How can you go from these basic laws towards an entity which is a thinking organism? And now, there's an answer to that question. The answer is one word: self-organization. That doesn't mean anything. but we understand what it means. We understand that it is by itself on the basis of the laws of our universe. Our universe, one can say, is structured in such a way that it has this possibility to self-organize. So matter has become complex by itself on the basis of how it functions. This doesn't explain anything, but at least makes you think that we will be able to understand it because we are the result of it. And you can even say that is a statement which is strong, perhaps it's a cosmic imperative. Our universe is built in such a way that, let's say, self-organization will occur. That's a strong statement again, but one of my colleagues, biologist, Christian de Duve, who got a Nobel Prize a long time ago, he said also life has happened as a cosmic imperative. It is built in, so to say, in our system. And also then, what are the fields which contribute? Chemistry is this bridge. And even a famous biologist, Arthur Kornberg, who is a very famous biologist, he said life is a chemical problem. And I think he's right. Of course, a very, very, very complex, with specific properties, specific features, but the basis, we are all built of molecules. So, the self-organization still, again, I come back to our friends, the physicists, they have not a unitary, unless you tell me that it has been found now, a single theory for explaining everything. So we have to look at it for 2 points of view right now. One is self-organization on the cosmic scale, as cosmologists tell us, and this happened through the action of gravitational forces on inhomogeneities which existed after the Big Bang, inhomogeneities in density of matter and of rate of expansion. I let them say that, that's their problem, but we are not, today we don't talk about that. What we talk about is the other self-organization of complex matter, which rests on electromagnetic forces, which bring together these particles, these entities which make up matter, and make them into more complex entities in a random way, but with some laws which make that this random evolves into a given direction. And also they operate on the pieces which make up matter, and then lead to this more and more complex structural combinations. So before there was any biological evolution, we all know about Darwinian evolution, but before that there must have been a purely chemical evolution. It's difficult to imagine, very difficult, that you jump from a particle to a living organism. And Darwinian evolution starts once there's an organism living. So there was a purely physico-chemical evolution, which progressively led to more and more complex states until you reach a threshold where you can get into something living. Now, how this happened, we still don't know. But in some ways, we will know someday. I think probably all scientists are quite convinced of that. So this pre-Darwinian evolution, in fact, covers also in some way Darwinian, because we will see, I mentioned briefly, that Darwinian evolution is also a chemical evolution, because an organism, when it evolves, changes its chemical structure. We can go back to that. So chemistry is considered, if I look at chemistry, as the science which deals with the structure of matter, how it is built up, and the transformation of matter, how you can convert objects into one another, of both living and non-living matter. In fact, I am a chemist because of this. When I was a youngster, doing experiments in the kitchen of my family, like many of us, many of us have started in that way. Huh? You get excited, you do your experiments yourself. Then when you realize that you can take molecules, you can mix them, and predict something and get something else. This is fantastic. This is, I must say, a power feeling. Huh? You act on matter. Come on. You have things, you mix, and you get. That is something really fantastic. That made me a chemist. So let's look a little bit at some historical milestones. I will cite a few, but I could have cited many more. But I cite the ones which I cite are nevertheless important. About 2,500 years ago, there were some people in a small country called Greece who, like probably also in other regions of the world, were thinking about what what is matter made of? What are we made of? What around us is made of? One of these was Empedocles, and he proposed that matter was made of 4 elements: fire, air, water, earth, and these combined into properties: dry for fire, earth; hot for fire and air; humid, air and water; cold, water and earth. It's an attempt. It is, of course, still very primitive, you may say, but okay, you start. But at the same time, more or less, there was somebody else had a much deeper insight. This was Socrates. I'm sorry, Democritus. Socrates was quite good too, but there was Democritus. And Democritus proposed that matter is made of pieces, particles, which cannot be cut to smaller ones. In Greek, that is atomos, atoms. Now, you may say, he didn't discover atoms, because I have to prove that they exist. But although the works of Democritus have not been written down by himself, but by his followers, his disciples, it is said that he based it on something which is, I think if it's true, it's fantastic. Because you can go into the street, at anybody in the street, you can convince them that matter is made of particles. How do you do that? You take a glass of water, you put a piece of salt. What happens? It dissolves. If matter is not the particles, but if matter is continuous, you cannot dissolve one into the other. So it is said that Socrates, on this simple idea that things can mix and dissolve intimately, said that, okay, this must be formed of particles. And I think if that's correct, it's a fantastic insight for that time. So that is sort of to say the pieces which make up matter. Then we have to jump because, of course, many things happen. Science has developed progressively. People have found that there are pieces. These pieces they have isolated. Chemists have done experiments, have isolated pieces, have put tables with pieces in it, but a bit chaotic. Nobody had an order. You couldn't see an order in that thing until in the middle of the 19th century came about Dmitry Mendeleev. Mendeleev found that, in fact, you can put order in these things which had been discovered. And he published in 1869 what I consider as one of the most important papers in science ever written. This one, on the relationship, it was in German at that time, on the relationship between the properties of atomic weight of the elements, the properties and the atomic weight of the elements. He had noticed that you could organize these elements which had been discovered according to their weight and to their properties. And he published this very fantastic table where he organized things, where he noticed that you can put, for instance, you can put together different elements, you can put them on top of each other, or you can put them in line and in columns. Even further, he found that there were some places, there was something not found, which was, he noted by a question mark. And he said, this will be found, and it was found. So this is one of the most fantastic tables ever published in science. It was a start. Of course, this table was not complete, but it had a lot of insight. So it is now known as the periodic table of the elements, and now it is like this. Now here again, I would like to insist on one thing. All the chemistry students have seen that. They must have. If you don't have, I think you better start your chemistry again. Because this table is absolutely incredible. This table has one fantastic property. It is complete. I mean by that, and as a scientist, I'm not very happy about it. I have to be a little bit dogmatic. I have to say it is complete. There's nothing else to put in. This is a table from hydrogen, helium, and so on. Why is it complete? Because it's just like arithmetics. 1, 2, 3, 4, 5. 1 electron, 1 proton, 2 electrons, 2 protons, and so on and so on. You just build up the atoms. And it is complete, which means also something absolutely incredible. Any matter in our universe is made of these elements. From the planet Earth to the end of the universe. It is these elements. I cannot escape it. I would like so to say, yeah, there are things to be discovered. But in this table, this is it. So if there's any living organism somewhere else, it's made of the same elements. And the carbon atom will always be a carbon atom. And the carbon-carbon bond will always be a carbon-carbon bond. It's incredible. So you have before your eyes, I think, one of the most fantastic advances in understanding our universe, our matter, ourselves. This table which shows you what everything is built of. I think usually I recommend to people to buy a nice one, put it above their bed. So every morning you can look at it. You say, oh, that's at least something we did, and you feel happy for the day. Now this is what? That's the playground of chemists. Chemists, they are like children using Lego. These are the Lego pieces, and how to play with them. You connect them, you disconnect them, you build small ones, big ones, and all that. This is making new forms of matter. Now, once you know what are the bricks, you have to know how you can change these entities into one another, transformation. That was first proposed and sensed by Lavoisier. Lavoisier wrote, nothing is lost, nothing is created, everything is transformed. Sort of saying that you have the pieces are there. They don't disappear or come back, but you can rearrange the whole thing. And this is the basis of chemical transformation, the basis of the chemical reaction, how you transform matter by performing reactions on the pieces of matter. At this stage, we know now that the structure, the composition, we know the transformation, but we still have to ask a question. Is it just a question of composition? No, there's something more, which chemists realized, the fact that these bricks are linked together in a given way. And they are linked in not arbitrary way. There's a given way. So around the middle of the 1860s, people had realized there is a connectivity between these bricks. And this connectivity can be represented by a graph, sort of a graphical way, like a picture. Many proposals were made, for instance, by Wurtz in Paris, Kekulé in Germany, in Bonn. But those two, they are not wrong, but they are quite far from what we use today. Loschmidt, a physical chemist in Austria, had much better insight. He said that carbon was a big circle, hydrogen a small circle, and this is, for instance, propane. And for a chemist, it is quite clear, CH3, CH2, CH3. It is not what we use now, but it was a correct representation. Even the oxygen had two rings, double bond was given, and so on. So this was big insight. And for instance, ethanol, a very important substance, as you all know, ethanol is this. We now write it this way, which is of course more clear, because we know that this is carbon, that is hydrogen, that is oxygen, hydrogen. But okay, it is something where we already see something very close to what we do now. One step remained to be done. We have composition, we have the way to transform, but we have the connectivity. But what about the shape? It was then realized in 1874 that these entities formed from atoms, these molecules, had a shape, an architecture. And this was proposed independently by two people, Van 't Hoff, who in fact the first chemist to get a Nobel Prize, who did many other things. And this, even what he published, what he published here was not accepted when he wanted to publish it. And Lebel in France, Van 't Hoff in the Netherlands and Lebel in France. These two proposed in 1874 that, let me sort of simplify, that molecules have a shape. There has a chemistry in space. They look like houses, like three-dimensional type of objects. And that was a very important step to realize that. Because on that basis also, another property came up, which will be the last I will mention, which is linked to the molecules of life, and that was the work of Pasteur. When Pasteur could explain that some molecules are related by a left hand and a right hand image with respect to a plane. And this property in Greek, hand is kairos, is chirality, the chirality of molecules. And this was, of course, Pasteur did many other things, but this was a very important contribution to chemistry. So we have now made a few steps along the way. I could, as I said, have said it many, many more, but it just gives you an idea. At that stage, we can say, all right, we have more or less have seen how from atoms you make molecules, and then you can, with that, construct what you may call molecular chemistry. Molecular chemistry, which is the way in which you link together the elements by strong bonds, covalent bonds, as the chemists call them. If we look at two milestones around the build-up of this ability to construct molecules, we can start with Friedrich Wöhler. In 1828, he synthesized a molecule, small one, simple one, urea, from ammonium cyanate. The name is not important, but what is important is that ammonium cyanate is an inorganic. That's something which is not contained in a living organism. Urea is contained in a living organism. So that was very important. What he realized, what he did there is for the following reason. Number one, it was a chemical transformation. Ammonium cyanate gives urea. Number two, it also changed even, I would say metaphysically, philosophically, our look at matter. Because at that time, people were thinking that you could not make a molecule contained in a living organism, like urea, which is contained in urine, without the help of a magic force, vital force. And Wöhler realized that by transforming ammonium cyanate into urea, this vital force did not exist. There's nothing like that. It's just a set of more and more complex molecules, and you can transform them, and you have a power on doing that. So that was a very important step. Now let's jump by 150 years or so to what chemists call a total synthesis. That means the construction of a complex molecules from its pieces, from its bricks. This was the total synthesis of vitamin B12, which is in our organism, a very complex molecule compared to the one we just saw. This you don't do alone. In fact, Wöhler didn't work alone either. But this was realized by the combined efforts of two groups, one under the direction of Robert Burns Woodward at Harvard, and the other one of Albert Eschenmoser at the Polytechnic School in Zurich. They collaborated across the Atlantic, and they were helped by 120, 150 men and women who, along 10, 12 years, went into this work to construct this molecule from the pieces progressively. I was postdoc with Woodward. That is my piece here. Now I show that not because just to say, it's my piece. No, because to show you that when you do science, you contribute to an enormous enterprise, and you bring a stone. I'm just a stone in the whole thing. Huh? Many people contributed. But it is interesting. You bring it, and nobody can take it away from you. It's your stone. Some stones are big, some stones are small, but it's your stone. So being a scientist is really participating in this enterprise. Now, since the 1970s, molecular chemistry has, of course, not stopped. Many new reactions have been found, ways of transforming matter, new properties of materials, new drugs, new processes for industry, and so on. But despite the fact that, of course, molecular chemistry is still a very strong chemistry and continues, one can then ask a question. Is there something beyond? Is there something else we should think about? This can be illustrated by starting with biological examples. This is a cancer cell, and these are killer cells. These cells, killer cells, are present in our organism. They are running around and trying to find out if there are some cells which went wrong, which were transformed, and they have to destroy them. They shouldn't make a mistake. If they make a mistake, either they don't kill the cancer cell, or they kill a healthy cell. No good. So how do the killer cells know that this is a cancer cell? Obviously, you can realize all this is made of molecules. There's a membrane which defines these things, and they touch. So something probably happens where they touch each other. Another example is that of a white blood cell with blue dots, HIV virus. When the HIV virus particle hits a white blood cell, it can infect How does the virus know that it now has reached the target? Now it can infect. Something again must happen between the particle of virus and the cell, which tells the virus, that's it, now I can go in. So there's a chemistry which has to be present at the interface between the objects, when they go together, when they touch each other. And this, beyond this molecular chemistry, there's another chemistry which we called supramolecular, which is beyond the molecule, which deals with what molecules do when they get together. When they get together, they like each other, they hit each other, they attract, they repel, and so on. And this is, let's say, building up a chemistry on the manipulation of the forces which are not covalent. Now, of course, physicists would say all that is the same physics. Of course, that is true. But it's for commodity, we can define, accepting that there are gray regions where we cannot make really the difference, but there is a domain which deals with covalent interactions, another domain which deals with the way in which things interact, sort of glue together rather than connect strongly. And although all those are the same features physically, they can be manipulated, in fact, in your brain and the way you think about differently. So this is then the chemistry of this non-covalent of chemists. They made their life easy. Everything that is not covalent, they call it non-covalent. So life is easier there. And this then led to the development of to the study of three features. How do molecules recognize each other? How can you transform them by their reactivity with one another? And how can you transport them to a barrier? When you have a barrier, like a cell membrane or an artificial membrane, how can they get through? And recognition is the basis of that. And this happens in our organism all the time. Without recognition, we are dead. We don't exist. So what is this molecular recognition? As we are sitting here, all your molecules, they do recognition all the time. If they don't do recognition, we are dead. We don't. Nothing, if you have short circuits, nothing works anymore. So molecular recognition, of course, first needs some kind of glue, some kind of binding. They have to touch. If they don't touch, if they don't interact, then they ignore each other. So there must be that. But that is not the only thing. If you want to recognize somebody in the street, you need to have information about that person, which means that molecular recognition means that molecular processes have information. They are informed processes. And all biology is based on information processes between entities which build up the biological objects. Now, a simple way, very simple, very trivial even way of sort of representing information is to say the objects have to be geometrically complementary, adjusting, and interactionally complementary. Plus attracts minus, plus repels plus. Very simple electrostatics, but there must be some basic rules like that. And the first proposal for explaining what we now call molecular recognition, which at that time was not called that way, but a very famous paper again in 1894 was by Emil Fischer, when he said, in that case he looked at an enzyme, when an enzyme acts on a substrate, they have to fit together like Schloss and Schlüssel, like a lock and a key. Of course, we now know the lock is some flexibility, the key has flexibility, can adapt, but basically, it's really true that you have like a key which fits into a given lock. So this is of course, a very, very important paper in 1894 for biology, for life, for chemistry. Now, let me just illustrate these recognition processes by what is arguably the most important recognition process for us living organisms. We all are the result of a genetic program, the genome. This genetic program is trivial. It is a long chain of rather simple chemical species, a sugar, a phosphate. And on this chain are fixed letters, just 4 letters, not more. And the letters themselves for a chemist is trivial. This is simple chemistry. Huh? 4 letters. Adenine, guanine, thymine, cytosine, AGTC. So, what makes a difference between a tomato and an elephant is this sequence. You have a long program, a long sequence, and you have letters, Okay, you change the order of the letters, you get something else. So this very simple program is the basis of the determination of what comes out of the development of an organism, the blueprint at the start. Since we talk about matter to life, let's look at one of those letters. Let's look at adenine. Adenine is simply a combination of 5 HCN molecules, very toxic molecule, as you may know, hydrocyanic acid. In interstellar space, this exists. 5 of them make adenine. For instance, HCN, this is a bit rearranged, but you have HCN, 5 HCNs make adenine. So you can very easily imagine that adenine resulted from combinations in interstellar space of HCN. If you add some water, you get guanine. You do some other chemical transformations, rather simple ones, and you get the 2 other letters. The conclusion of that is that the living matter is made of basic building of, what determines the nature of a living entities is based on a program which is a simple string with letters in a given sequence. Now that's the storage of the information. How do you read it? The reading is also very simple. It happens that adenine can undergo a 2-point interaction represented by these dots with thymine or uracil, it's a bit of transform here. Guanine has 3 interactions. So 2, 3, 2, 3 is the way you read it, to read it. In other words, the storage of genetic information is a chemical string with letters on it, only 4. And the interaction 2, 3, 2, 3, a binary system, a binary process, is the way to read that information. Now you can wonder, why is it not more complicated? The simplest answer is that it's enough. 4 letters, 2 combinations, that's enough to do it. It is quite possible that in the course of evolution, more complex systems had evolved. But probably it's a mess. You get much more complicated. It gets too complicated. So evolution has probably taken what is enough to get forward. It's rather strange that such a simple system is really what living organisms depend on. So that makes me to again say a song in praise of chemistry, which is that chemistry stores information in molecules and processes information in the supramolecular way by contacts between molecules. So chemistry is also a science of informed matter. And this is perhaps the most important conceptual aspect that supramolecular chemistry brought to chemistry. Of course, it has interaction with that, but the fact that chemical objects carry information, and the information is read by the way they can interact. Now, just for fun, this is a nice double helix. See, that's a nice double helix. You can even say there seems to be base pairing here. But there's a picture which you can find in a book. And that's an earlier representation of double helix. Of course, the person who drew this picture didn't know anything about DNA. But furthermore, what is very strange and interesting is that to transfer DNA, what do you need? A man and a woman to make a baby. So that's not only the storage of information, but also the transfer of information. So just a small word about how we scientists often function. We often start with an idea, an interest we have, and then this interest we study it, and then we are led to many other things. And so just let me tell you how it started for me. When I came back from postdoc, we did mostly molecular physics for some time, because I wanted to do not my work for PhD with Guy Risson, a great French chemist, not with the work of postdoc with Woodward, but something else. So that's why we did molecular physics at that time. But then I was always tempted by, of course, biology, and especially by one thing, which, of course, makes us, how does our neurosystem function? Then when you are a chemist in 1965, 66, studying the neural system looks like impossible. So I thought, okay, let's see if we can study a simple process present in the nerve system. And the simple process accessible to a poor chemist is the propagation of nerve influx. That sounds complicated, but in fact what is simple is that it rests on what is called the action potential. The action potential, which transmits information along the axon of nerves, rests on the change of concentration of potassium and sodium ions through nerve membranes. They propagate, this impulse propagates by changing the concentrations of sodium and potassium across a nerve membrane. So that's okay, sodium, potassium, I know what it is, that's something maybe accessible, so maybe we could start with trying to understand how we can handle that, how we can perhaps modify, transport, or make systems which do that. So can one make synthetic molecules which would perform a discrimination between sodium ions and potassium ions? Like in a nerve membrane, there must be molecules able to do it. And this led us to go back to good old Emil Fischer, of course, which is to say, okay, we have a bunch of keys, we have a lock, and if we have a number of keys, this one, the red one, will fit. So can we do Emil Fischer type of complementarity in other type of systems and build up progressively? Many, many people have been looking at that over the years in China, in Hong Kong, in many places of the world. And many studies have been performed, also because you will see later there's also biologically that very important. Now, when you start a project, it's better to look for a simple approach at the beginning and then make it more complex. So if you think about periodic table of the elements, a sphere is the simplest object in three-dimensional space. So the spheres in the periodic table are the alkali cations. Lithium, sodium, potassium, rubidium, cesium. A very nice set of balls with one charge which increase in size. And among them, sodium, potassium are, of course, what I said. They carry the nerve influx. They're important in the blood. They're important for heartbeat and things. So you sort of ask the question, can we develop molecules which selectively bind and transport these entities? This would be a spherical molecular recognition. So long time ago, in 1966, we made these molecules, which we call cryptans, because the crypt has a cavity inside. These are synthetic, made in the laboratory with cavities here. That is the lock, cavity, 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 increasing in size. You can do that by synthesis. And this one fits lithium, this one fits sodium, this one fits potassium. Now maybe I should tell you also that those were made in my laboratory in 67, 68, by two people, Bernard Dietrich and Jean-Pierre Sauvage, who got the Nobel Prize last year. So we were very happy about that. And so this is the beginning of these recognition processes. And of course, lots of more complex molecules have been studied. I don't want to get into it because, as I told you earlier, any biological process is a recognition process. So that is just really the ground zero level of recognition. But it's the start. And we studied then more and more and more complex recognition processes, and many, many laboratories joined the game. Just to show you that also chemists are interested in the aesthetics of the molecules. This is again the one I showed you earlier. And here is another nice representation. That's a sculpture where you have made for me by Bela Vizi, a colleague from the University of Veszprém in Hungary, chemist turned artist. That's one of those cryptates. And he has a nice inspiration. And I show you four more of the sculptures he produced, these four. He made many more. But I want just to show you one, this one. It looks very nice, isn't it? Like a perfume, like an odor sort of developing. In fact, it is called the scent of the rose. Now, for anybody, you see scent of the rose. Okay, it looks like a scent, sort of perfume developing. But for a chemist, it's perfectly understandable. The scent of a rose is dominated by one compound, phenyl ethyl alcohol. Phenyl ethyl, CH2, OH, alcohol. So for chemists it's perfectly understandable, but it's actually also nice. In fact, one other one, this two, is linalool and acetate linalool, the two dancers. And this is two chemicals, these are two natural products, these two, and there are some others. That's quite nice. Molecules have also some aesthetics. So let me just summarize at this point. Molecular chemistry was defined, let's say, by the work of Friedrich Wöhler in 1928. Then you use this synthesis, which is developed on that base, to make what you may call a lock, a receptor. And then you try to see beyond that, supramolecular chemistry, which the first time I used the word was in 1978. And then you can study the interactions between the entities, and then you want to sort of use this to develop understanding of recognition, of transformation, and of translocation to make what you might call, functional supramolecular devices. And again, as I already said, many people have joined this game. So a few applications. One is usually asked about applications, but I want to illustrate them, first of all, by applications indeed, but also by how difficult it can be to get to an application. It's quite obvious that molecular recognition is absolutely fundamental in drug discovery. A drug is a key for a biological lock. You want to make a molecule which is as specific as possible to attach, to interact with a biological target very, very specifically, and not to make a mistake. And this is the basis of discovering new pharmaceuticals. So I don't want to say more. That's sort of evident. We also developed a method for medical diagnostics, then a word about gene transfer and about biomaterials. So another type of cryptate we made for other reasons, to understand some photophysics, was to make this one, which had, again, an organic shell. And in the shell was a europium ion, one of those elements of the periodic table. When you put it in and shine UV light, the energy absorbed by the organic shell is transferred to the europium ion, which emits red light. So you can use this thing and attach it to an immunomolecule, a molecule capable of recognizing proteins and specific targets, epitopes, and you can develop a medical diagnostics machinery, an apparatus here, thanks to the hard work of Jean Matisse at a small French company, which he has now changed and so on. It's now called Cézanne. Is in Nîmes in France. And this led to this immunoanalysis system, which is now used in many hospitals for various methods of detection, of also drug screening, also for companies to have methods for high throughput screening and things like that. Then another process which I had studied over the years has to do with gene transfer. Gene transfer is a very important process because it is related to how you can modify the genes. And we had studied the basis of transport processes for some time, and then we had sort of a little bit dropped it because it had quite a lot of work, until came back this fact that you might use gene transfer, artificial ways of doing gene transfer for biotechnology, for gene therapy. And at that time, the process for a chemist was to make a synthetic vector which then would carry DNA into the cell. There are other approaches, viral approaches like Alain Fischer did, very powerful because they are viruses, they know how to do gene transfer, but you can also do it less efficiently, but with other advantages maybe, by synthetic vectors which sort of make a shell around the DNA, which is a polar molecule. It doesn't want to cross the cell membrane, so that this can now go into the cytoplasm and transform all the machinery, very complex machinery, and the output is a given protein. So this gene transfer is a very important process for biotechnology and gene therapy. At this stage also I usually make a remark about genetically modified organism because that's genetic modification. And I say that genetic modification of organism is absolutely necessary. And when people say we shouldn't do it, they're totally wrong. Not only have we done it, we will continue doing it, and we need it. It's a way to control what we have done all the time. When you eat an apple, it's not a wild apple. It has been transformed for many, many, many years. Of course, we will see naturally. Of course, you mix all this genetic information together, and often you don't know what you're doing, because you just mix and you wait until you see the result. When you can modify the gene precisely, you can induce a given property. So, of course, one has to be careful. But genetic modifications, genetically modified organisms, are a necessity for mankind. What about materials? There something interesting came up which had to do also with how long it can take from a discovery or from a conceptual proposal to an application. It happened that in 1990, in my laboratory at the Collège de France, we developed a class of polymers which was called supramolecular polymers because they are linked by non-covalent interactions. So this worked and developed. 1990 was the first paper in the theme, and then it developed. And then suddenly, in 2013, I got an email from a small company telling me we have made biocompatible polymers of supramolecular type, and they have been used for cardiovascular implants, especially for treating children who have a cardiac malformation and need reconstruction. So on the basis of these materials, for reasons which I cannot get into, they made that, and it was introduced into this. The first one, Dominika, a little girl, Russian, because the implantation was done by Leo Bokiria at the Bakulev Center of Cardiovascular Research, Surgery. I didn't know Bokiria, I now know him. He looks like a surgeon, doesn't he? Strong man, huh? You have to be strong. This little girl, this is in October 2013. 3 months after implantation, she looks very fine. She still is very fine. And many more children have been implanted, more than 10 now. This is a slide was made some time ago, so made of 10, 15. They also made now heart valves. You don't need any more heart valves from a pig or from another type of polymer. So this took 23 years. So it takes time. Sometimes it's much quicker, and sometimes it takes a long time, especially when it is biomedical, because you have a number of steps you have to go through. You have to find that might be interesting. You must have the courage to develop it. You must find the finances for doing it. You must find the surgeon who is ready to put it in. You have to go to all the regulatory steps. So it's a lot of time, but eventually people are able to get there. And of course, we're very happy about this result, knowing that the children running around with some supramolecular polymers in their heart, that's a very great satisfaction. Now, another process of great importance is supramolecular materials can heal themselves because the interaction between the entities are weak and can reform. This is a nice film made by an Indian postdoc in my group. You cut it, you see it's a very nice transparent film. You cut it with scissors, you superimpose the two ends, and you press with your finger just a little, and you can stretch, and it sticks again, almost as well as before it was cut. So this is just one example, and there have been many other people working in it, for instance, Ludwig Leiber in France and some others. And this is a very important property of supramolecular materials. So at that stage, let's go back to this line of self-organization. Beyond pre-organization. Pre-organization is what chemists like to do. They have an idea about how to organize matter, so they pre-organize it for a given feature, like making a lock for a key. But then you can say that, okay, when I understand that, I perhaps will be able to make systems where the components of that will find the way to organize by themselves. And this is done trying to set up in the laboratory molecular recognition directed self-organization processes where it's spontaneous, but it is along a design. You design your thing, you put information in so that it assembles in a given way. I just give you some examples of that. First of all, a biological example, because we are self-organized, and a virus is the simplest case of just at the frontier of living matter. A virus is not living. It needs some machinery of the cell to reproduce itself. But take this tobacco mosaic virus. Tobacco mosaic virus is a tower, helical tower, made of 2,130 bricks, which have a given shape, And where the surface is, it happens that this brick is self-complementary. One side is complementary to the other side. But the shape is such that, like this piece of pie here, which you can't get together, and they get together and they self-organize. And so this, when you mix these particles, when you take this unit, you mix them, you get towers of all kinds of lengths. Then you add the RNA, the genome of the virus, and then it gets localized to a given entity. It now forms the viral particle. Because they touch here, so when there is nothing to touch anymore, they stop developing. So the virus is a good explanation, at least good illustration, of self-organization of a set of molecular entities which are not yet living. In fact, I digress a little, but okay. There's an interesting movie you can find on the internet made by students or graduate students from California, which shows that this magical thing, getting things, organizing things, is in fact sort of trivial. There are some viruses which are spherical. They are made of molecules which have this shape where they can form a sphere. So what these people did, they took plastic pieces, centimeters big, and they give them the shape of this sort of piece of a sphere. They put little magnets at the points where these proteins interact. They put them in a beaker and you shake it. And after 2 minutes you have a sphere. It looks like magic when you look at it, but obviously it's trivial. It means that it is more stable to have an entity like that. They even took red-colored and green-colored objects with a little bit of different size. Then they use a machine to shake because it takes a bit longer, it takes 20 minutes. But you see this thing growing, sometimes a green piece with the red, but then this falls out, and at the end you have a green sphere and a red sphere. You can find that on internet. It's really worth looking at. It's very impressive. And you realize that, okay, self-organization with molecules, it's not so complicated and not magical at all. So this looks like sort of a programmed chemical system. The program is molecular, and its operation is supramolecular. And you enter information, and you let it go. So let me just illustrate that by, there are many ways of approaching it. We have first started this in the following way. If you use as brick an entity which is an organic molecule able to bind to a metal ion, coordination chemistry, and you use as cementing between the bricks the metal ions which bring them together and link them together. If you design correctly the ligand, this binding molecule, and you use the good, so to say, binder, cementing unit, you can make things like that, for instance. That's a natural double helix. That is double helixes we made now in 1974. This is a double helix. This is one molecule. That's the other one. You don't see the middle ions. There's one here, one here, one here. They're a bit hidden behind. That's a triple helix with here nickel ions. It works also with iron. You can make triple helices. And you just, of course, you have to make the molecule. But once you have it, you mix the midline and you let it go. And you build up. More complicated object, for instance, this one, a nanocylinder. These are sort of straight molecules, linear. These are flat molecules. And these little golden spheres are copper ions, copper 1. You mix 3, 4, and 12, and you get automatically 99% of this object in one shot. This is just some examples, but since then, many, many more complex entities have been formed of objects which a synthetic chemist proceeding the stepwise way, we said, you are crazy, you will never get it. But it works if you handle correctly the way you design the brick and the way you make them interact with metal ions. These are some others which look nice. A grid and these objects. Of course, computers allow you to give nice colors. Of course, the molecules are not colored. But these are just to illustrate what can be done. There is of sort of some interest also, maybe more than some, for nanoscience and nanotechnology. Why? Because the present approach to all our fantastically complex objects, like this little computer here, is very, very powerful, no doubt. But you may say, maybe there's a combination with the properties of matter to self-organize. because eventually, rather than having to make the things, maybe you can devise a way that they make themselves. And this would then be self-fabrication, which is the ultimate way to fabricate. Maybe, if I want to be simplistic, a good proof that it is possible is that our brain is the best computer and it is self-organized. We don't make it. So it's possible. So self-organization, I illustrated. I want just to show you now a little, just for one slide later, to show you what is now the evolution of our work. So a bunch of it is to design the system, to manipulate it. This is a way to understand how you can do it. To design, to put given shapes of molecules, given connectors, and let it develop. But then the question is, can you do it with selection? That's another step in complexity, where the system would select from a diversity of possible bricks the ones it needs to build itself up. You need also dynamics, because you don't want the first collision to stick too strongly, because then you are finished. You may be in a wrong valley of your hypersurface of structure and energy, but you want to get out of it and search further. So you need dynamics. This then leads to the fact that you would like to develop a chemistry where the constitution of the chemical object is dynamic. I mean by dynamic, which can fall apart and reconstitute itself by different shape, or by incorporation of other pieces, or by expulsion of throwing out some others. And this leads to now a property with our main line of research now is adaptive chemistry. Can one make systems, can one make chemical systems which can adapt to changes which you force on them? These changes can be physical, temperature, pressure, heat, and so on, and others, electric field, for instance, or a chemical agent like a protonation, like a metal ion, like another molecule. So can one make adaptive systems? Just to indicate what this could be good for, this chemistry which is dynamic in the constitution of the object. Like if you have a house and you take out bricks and put other bricks in the place, this can then be of interest, as I said, for adaptive systems, and which then has been used for new methods for searching for biologically active substances, for having nanostructures which pick up the pieces they need to build themselves up in a mixture of pieces, and also for dynamic materials. An interesting notion there, which often in science you have to get away from what you have been thinking about all the time. For an organic chemist, mixtures are something terrible. You want to have pure substances. But then you realize, and for biology it's obvious, a cell is a horrible mixture of many things. So chemist has to go the way to work with mixtures. When I did my PhD, when I had a mixture, you put it down the sink because you cannot analyze it. Nowadays we have the methods. So now we have to work with mixtures. We have to understand how mixtures behave, how a system can be programmed well enough that despite the fact that it's a mixture, it still can form. So there are very interesting way doing chemistry with mixtures. When you talk to a graduate student, PhD student, or that, they look at you guys and say, what is this guy telling me? He wants us to work with mixtures. Say, yes, but mixtures, that is what we are. We are mixtures. Our cells know exactly, the molecular cells know how they want to interact, where they want to interact, at which rate, in which position in space even. So there are a lot of very interesting, that's a fantastic, a very exciting field. So, evolution of chemistry, we start with molecules. We all have to know molecules first, because that's the basic building block. Then they can be adjusted, and so that they interact in a given way. Then they get organized by themselves, because that's the way they interact. So let's say, for being a bit more mathematical, the algorithm for reading this program is the way these things interact. It can be this way, it can be this way, it can be different ways of interaction. Then dynamics, you need dynamics to be able to explore adaptive, and then more and more steps towards living matter. Now, this being said, I would like to make some general remarks on chemistry and science in general. First of all, chemistry is not just to discover what already exists, but to create what does not yet exist. For instance, the book of chemistry has to be written. You have to read it, of course. You have to read what already exists. But you can also write it. Like the score of chemistry, you can compose it and not just play it. So chemistry has creative power, which again I want to represent by one of my favorite sculptors, Auguste Rodin. Here you see the hand of the artist expressing out of the stone a sculpture which is not contained in a stone. It's the hand which makes it. The stone is a stone. And this is what chemistry can do also. Out of matter, you can create objects which did not exist before. And I think for youngsters, that is something fantastic. You always think about the fact that you can create new entities which did not exist. Picasso made paintings, of course you can see them, but you make a new molecule. It's a piece of art, so to say. It's creative. But obviously, it's not very attractive for people because it's a white powder, so what? Now, this possibility was already sensed, of course, not in the same terms, by somebody who is at the same time a scientist, an engineer, and of course an artist. Leonardo da Vinci, he wrote a fantastic sentence, where nature finishes to produce its own species. We are part of that. Man begins using natural things. That is now, we know, the periodic table of the elements. With this, in harmony with its very nature, the laws of physics. I can't get against that. That is control. But then the ending is fantastically strong, to create an infinity of species. This is really quite fantastic. It's not just chemistry, but it's this possibility with science to create things which did not yet exist. Now, again, coming back to this, there's some Chinese, I cannot read the Chinese. I've asked one of my students to write it for me, so I hope it's right. In the ancient Greece, there were the gods, and there was some guy, Prometheus, who stole the fire of knowledge from the gods. And he ran away. You see him looking over his shoulder to see if they don't run after him to catch him. But he succeeded in giving it to mankind. Now, this is very important because then mankind could develop science. Here is the light of science. One important thing about it is knowledge, we cannot give it back. What we acquire, we have. You cannot wipe it out. Knowledge is there. So we have to learn to live with it. And we, by developing science, we look for knowledge. But this knowledge, you cannot give it back, but it will allow us to control our own destiny. I sort of think that it's now us to make evolution happen. Evolution has been the statistics, the random evolution of living organisms. And now we can do it. Question how and why. Okay, these are discussions you can have for hours and hours. But things will develop, and in 50 years, I would like to see, I will not see it, I would like to see what has happened to it, how mankind has been able to handle this power of knowledge. Please, I hope it's correct again. There is Hanyu, I like this very much, for researchers also. When you sit at the bottom of a well, the sky seems very small. You have to stick your head out, look around, and see what all the beauties which remain, which you can see, the ones which remain to be discovered. In the past, we transferred education, information in this way. Presently, this way. Do you know what that is? Zip it. Yeah. Michelangelo. Now in the future, what could it be? Maybe this. Now this is only a way to say the first step in the way in which we will transform each other, it has already happened. We will put in our body artificial pieces. You may have new teeth. That's not your tooth anymore. These are new teeth. These are ceramics you put in. You may have a new lens in your eyes, and that helps a lot. But this is artificial. These are polymers. You can have a hip with some titanium. That's artificial. Heart transplantation is a bit different, but it's the heart of somebody else. Is it still you? You have heart of somebody else? Is it still you? Are you you? If you say to your wife, I give you my heart, it's not yours anymore. No, it's really some fundamental questions you can ask yourself. So this will happen. I'm not a fan of going too fast, but I'm convinced that we will do it because we have the power to do it. It will happen probably in the future not by this material insertion, but probably more biological, by stem cell control, stem cell control, development control. And you know, I would almost have become a molecular biologist because in my first lectures at university, we had a course on the development of the egg of a salamander, something like that. And that was work done by Hans Spemann. Spemann did that in the 20s. He got a Nobel Prize for that. And this was fantastic. The professors said to us, look, we looked at this development. If you want to see the next step, come to my next lecture. Everybody came. Because what happened, Spemann had shown, if you take an egg, you know when it develops, that certain regions of the egg will give the heart, the neural system. And so he had taken a piece of the cells which will give a heart and put them to the other side. And the piece had two hearts. Of course, one was functioning, and the beast didn't live very long. But he had shown the first time what now Yamanaka and the people who do stem cell work have now been able to do, to show that it is a mechanism which is based on, in fact, molecules, because they interact. Yet, and Yamanaka has even shown that if you take a skin cell of a mouse, you can make a mouse out of a skin cell. That's quite something, no? A skin cell contains all the information. What you have to do is to deconstruct it, to give it back to stem cell and let it develop into a mouse. In the future, maybe you don't need to have heart transplants anymore. You don't need a donor for a heart. You can make your heart. You put it in the fridge until you need it. Okay, that's science fiction in some ways, but it's not so fiction because it might be possible to do so. So I want to finish with a mathematician, not much about mathematics right now. So David Hilbert is a famous, very, very famous mathematician. He looks nice here. Huh? He's buried in Göttingen. And he has a tombstone here, and he has two lines written on his tombstone. What is it? Very, very interesting for scientists. Wir müssen wissen. We must know. Second line, wir werden wissen. We will know. That's very strong. But I think that's sort of all that drives us too. So, with the help of science, we will know, and all the young people here, and the older ones too, science shares the future of humanity. It shapes the future of humanity. Please participate, all of you. Thank you very much.
Yeah, thanks, Professor Lehn, for the very inspirational talk. So now the forum is open for question. Any question? Yes? From the back? Yes. Oh. Yeah. Maybe I can give this to you.
So, Professor, thank you for your lecture on more likely a historical view on chemistry. So, my first question is that you talk about cosmology is very different from chemistry, but I personally feel that it's like it's in quantum physics there is corresponding principle. Do you view that
what is
corresponding principle? It's like talking about when you're in infinity and in the first level of energy level, and then there is a huge difference between classical mechanics and quantum mechanics. So do you view like that? And also, you talk about that there is four elements in the DNA on the DNA helix. Do you view that because organic chemistry always talk about chemistry equilibrium? Do you think that sometimes? Do you think that because of pressure and temperature, this sort of factors that is also affecting the combination that hence create the complexity of DNA, and also is that nowadays education is very divided in science field because we are so divided in biology, chemistry, and physics. In some sense, should we divide over every subject or we should learn everything? So this is my free question to you. Thank you, Professor.
Wow. That's another talk.
A lot of questions. Yeah.
What I have tried to show that progressively complexity has built up, and it happened on the basis of the way in which molecular objects form. And of course, molecular objects are based on the way in which elements interact, and the elements exist because of the basic laws of how particles get together. So you had the second question then was about equilibrium. Now, it is clear that what I showed you is equilibrium self-organization. For more advanced lectures, I discussed that a bit. Life is an out of equilibrium system. We are all out of equilibrium. So the next step, a very important step, is to introduce non-equilibrium states in what I showed. We have a few. There are other people working on trying to have systems which are out of equilibrium. The most simple way to get that is by photochemistry. You shine light, you have a shape of a molecule which is not thermodynamically anymore at equilibrium. You go a higher state, and then you observe what happens when it goes down to equilibrium. So combining self-organization system, this equilibrium self-organization I have shown. What I showed was equilibrium self-organization with out of equilibrium states. That is something which is now in the process. And we have a case where on the way to another object, there's a first one being formed. And in that case, it is slow enough so you can observe that you have an intermediate and then the final one. But of course, this could be observed because it's slow. It is still a simple way. The real way, the way in which we must in the future look at it is to combine the organization to, let's say, flux of energy, of temperature, to systems which are out of equilibrium on the physical side, like Prigogine type of systems. But Prigogine is sort of, let's say, it's an operational way of describing the objects. You have to make then molecules and couple it with reactions. Cells do it, so it works. But we have to see how we build it to build this up.
Okay. Do we have any other questions? Yes.
Yes. So I will ask my question in English. You did not mention much the power of computers in all this field. And right now there is lots of progress on machine learning, and neural networks optimization and so forth. So could you comment a bit on what you see in this big program, how the matching of very clever ideas about these structures could be also developed by computers and self-learning computers?
Absolutely. First of all, of course, I did not mention computers, but it's obvious that now there are programming systems. So it's Martin Kaplus, when he got the Nobel Prize for molecular modelizations. These are now powerful programs which can allow you to analyze a given system first before you make it. At the end of the game, you have to make it still. We use it also, but I have had a career in that, so I can imagine many molecules, the way they probably will behave. I cannot put numbers. A computer puts numbers on it. And this is a very important thing. Now there's another aspect in this chemistry I mentioned at the end, this dynamic chemistry. You get mixtures. And in these mixtures, if you touch one of the constituents, it will influence the others. So I hadn't time to talk tomorrow, no, not tomorrow. But this is a network. So in a mixture of exchanging pieces, you have a network of interconnections, where some are agonistic, others are antagonistic. So with simple networks, you can still represent it graphically. But with more complex ones, then you need mathematicians working with networks, neural networks, and so on. So that's another aspect. Learning, we have a case which we just finished recently of molecular systems which learn. Learning is an out of equilibrium case. When you learn something, it's not thermodynamically at equilibrium. So we have made a system where the transformation is a dynamic system, but where it's not fast, but slow enough. So that you have this system, you put in something, the system will adapt to what you have put in. And then you take this thing you have put in, you take it out, and the system will still have the same composition because it's slow. But then if you wait long enough, it goes back to equilibrium. So that's a very first step in what you might call learning. You impose something, then the system learns, it changes itself, and this learning is kept, so it's a memory. You take it out, you still keep it, and when you add it a second time, the system is ready to recognize what you add. So that's a very primitive way, but progressively you can make chemical systems which have this, some of these features, very simple still, which define learning. But it's true that, I mean, we always, there are many people who are, for instance, Hopkins and neural systems, Hopfield, pardon, has neural networks, and okay, there are a lot of things to do. We can't do everything. We'd like to. Physicists have to help mathematicians. We have to get together. It's not always easy, but the Collège de France is a place where you can talk with people from other type of fields. I think we don't do it enough, even at the Collège de France.
Great. Any other question? One last question? Yes.
Thank you, Thank you, Professor. When we talk about the matters and life, how will you evaluate the work from the physicist Erwin Schrödinger? Thank you.
Oh, he wrote a very interesting book about what is life. But it's a very interesting book. But he didn't explain things. But it's good. Like when you have a theory of the ideas about a complex system, it's interesting book. But of course, biology will tell you that there are many other ways. There are a lot of things behind it, and the fact that you say it, that you write it and describe an idea, doesn't mean that you understand the way it really proceeded. But if, for instance, since you speak of Schrödinger, of course, the Schrödinger equation, in principle, if you could solve it, of course you can now push it to the ends, would give you organic chemistry, molecular chemistry, superorganic chemistry is just there, sure. But you still have to make it. But in principle, it's lying there. But the principle is not enough. Like making a drug, you can design the drug. But if you want to take it, you still have to need the material. Otherwise, you have nothing. You cannot just look at the computer and say, oh, what a nice drug.
Okay, great. Please join me to thank Professor Lehn for the very inspirational talk.